By now, you already know this. I'm going to spare you all of the things that are on the slide, but the WESTalks is brought to you by IC Impacts and UBC Future Waters. And this is the wonderful organizing committee that's been wonderful to work with. And we've been hosting this for the past 1 year. There's myself, Abhishek, Layli, Fuhar, Carl from UBC, Jaskern from the University of Guelph, and finally Feria from IC Impacts. If you already haven't made sure you enter where you're actually from, please click the link that's being posted in the chat box so that you can tell us where you're actually from. And we are always excited to see where our audience is coming from. So it's been great to see the coverage that we've had across the world over the past 6 months or so. And finally, we've already had a wonderful lineup of speakers. And today we have Dr. Frederik Hammes from EAWAG. And the West Talks is going to continue up until the West Conference. And we have 3 more talks lined up until then. Before I introduce today's speaker, just a reminder that if you did miss out on any of the previous talks, you can always log on to YouTube, the IC Impacts YouTube channel, and catch up on the previous talks. And finally, I'm very happy to introduce Dr. Hammes. I love that he was the only speaker who had a black and white photo. I thought it was quite cool. And Dr. Frederik Hammes is actually a group leader of the Drinking Water Microbiology group at the Swiss Federal Institute of Aquatic Science and Technology. He has a bachelor's degree from the University of Pretoria in South Africa and a PhD from Ghent University in Belgium. His research group currently focuses on measuring, understanding, and managing the microbial ecology of drinking water, with current emphasis on opportunistic pathogens in building plumbing systems. Before I hand it over to Dr. Hammes, just a reminder that if you have any questions, please leave them in the chat box, and Layli will be getting to them at the end of the presentation. And looking forward to your presentation, Dr. Hammes. Over to you.
Right, thanks a lot. You can see I got invited in the middle of a pandemic with that photo. All right, I'm also just going to post here in the chat for you guys the papers that I will be presenting about today, if you want any of them. And so with respect to the length, I'm going to run, I think, around 30 minutes. If I'm over time relative to your own schedules, just shout out and make me stop. That is no problem at all. I can end at any good time in this presentation. All right, let's see if we can get this up and running. So that should be working. So good afternoon. No, that's good morning for you guys, everyone. Thank you very much for the introduction, and thanks a lot for the invitation to present here in what is a really super seminar series. What I'm going to do today is talk a little bit about our research during the last couple of years on the microbiology of building plumbing systems, and specifically focusing a little bit on how consumer choices and engineering decisions influence the microbiology in our houses with us. And so to build this talk, what I'm going to do is I'm just going to build it around these 8 publications, which I put in the chat for you. And I'm going to try and argue that, building plumbing microbiology is interesting and relevant. And then I'm going to take you through a couple of studies ranging from full scale all the way down to lab scale studies, where we try to understand a little bit better what's happening in building plumbing systems. And then in between, I'm going to try and tie this up a little bit with respect to management and how we can control the biology of building plumbing systems. But already not to spoil this, but there's no golden answer at the end of this talk. This is a slow process building up. And so before I actually jump into actual pipes, I would like to talk about rubber ducts, because I think rubber ducts is one way in which one can understand quite easily what's happening with materials in contact with drinking water. Whereas if we talk about pipes, they're often hidden behind in the walls of our homes, and it's not so easy to understand this. And so some of you may view a rubber duct in the way that you see this one on screen. I prefer to look at rubber ducts in this way. So what I can highly recommend, if you've never done this and you have a duct at home, slice it open and enjoy the biofilm that is forming on the inside of this duct, which you can see very clearly in this image over here. And so slicing open a rubber duct tends to get you a little bit of attention. And so no matter whether you're on the side of thinking rubber ducts might kill your kids or whether you think they won't, I think from a microbiology perspective, we should rather appreciate the biology that forms inside of them and try to see what we can learn from this with respect to how these biofilms form on materials in contact with drinking water. And so this picture over here, this is just a beautiful electron microscopy image coming from this rubber duck, which you've seen just in the picture in the slide before. And you can really see the biofilm super nicely in there. And so I'd like to talk a little bit just for this introduction about how to assemble a rubber duck microbiome, how to build biofilms inside rubber ducks. And so if you want to build a microbiome, the first thing you obviously need is microbes. And in the case of rubber ducks, where these microbes come from is obviously from the water in which the duck is swimming. And so in Switzerland, that would mean about 100,000 bacteria per milliliter and about 5,000 taxa, give or take, that is in our drinking water, which would then immigrate into the rubber duck. Now, here comes the first very important consumer decision, because you as a consumer can decide whether that duck is swimming with you in your dirty bath water or whether you let it swim in the clean water before you get in. And that's going to have a tremendous impact, because if the duck swims with you in the dirty water, then of course it also gets immigration from your own skin microbiome and maybe some of the dirt which you're washing off in the bath, and that contributes to the dispersal of bacteria into the rubber duck. Now, the second thing, if you want to build biofilms, you need food. And here, this is a very clear engineering decision. Whoever decided to build this rubber duck decided on a type of material, and this plastic or synthetic material is going to leach organic carbon out of the plastic into the environment which the biofilm will grow on. And so you can expect a lot of organic carbon in the range of 4 microgram carbon per square centimeter per day, which is quite a lot. But again, this is not only engineering decision. You decide if duck swims with you or without you in a bath. If it swims with you, it's going to be exposed to shampoo, it's going to be exposed to soap, it's going to be exposed to bodily fluids, which means additional organic carbon, nitrogen, phosphorus, all those things that helps to build a microbiome inside this duck. And then finally, if you're a good parent, you're going to think seriously about maybe also cleaning your duck occasionally. That's a consumer decision. You're going to expose the duck to high temperatures and to chlorine, the very same thing we do in our drinking water sometimes, and that is going to affect the microbiome or inside of this duck. And so the long and the short of this is what you have is these two ecological processes of dispersal and selection, which is influenced by consumer decisions, it's influenced by engineering decisions, and that will in the end result in differences in the amount of bacteria and in the composition of the bacteria inside of these rubber ducks. All right, and so that brings me slowly towards a little bit towards building plumbing. But before I get there, a question that I often get is, so we have these organisms or the biofilm inside the ducks, and is this really such a problem? I mean, do we really care? And I think a point I want to make very clearly is that most of the microbes also in these ducts and in the pipes and whatever are completely harmless, and in many cases, I believe they're even beneficial. However, there are some cases where problematic organisms develop inside rubber ducks and they make children sick. This has happened. There are also very clear examples where problematic organisms develop inside our building plumbing systems and they make people sick, and occasionally they kill people. And for that reason, I really believe it is important that we understand better what drives the development of these organisms and how we can utilize that to control them in the future. All right, and so if we look at a drinking water system from source to tap, and I really enjoy the entire system, but for the last couple of years, my group has been focusing more and more on the last bit in this picture, so on the building plumbing system, and specifically going towards the last meters in the system. And the reason for that is that is where a lot of very interesting things are happening. So for example, if you consider these two parts of the system, the larger distribution system outside your house is publicly owned, and of course the building plumbing systems are privately owned. which means you have much less access to the systems for collecting samples and for understanding how they are operated and things like that. Then, of course, there's very clear regulations when it comes to public drinking water distribution systems and governmental control and things like that. Whereas when it comes to buildings, there are regulations, but it's much less. And I would call that literally the Wild West when it comes to good regulations. Then there are many scientifically studies done during the last 2 decades on larger distribution systems. There are much fewer studies done on building plumbing systems for the very simple reason that, for example, they're so difficult to access due to privacy reasons. And then finally, and this you can argue with me, but I view larger distribution systems as relatively stable environments opposed to building plumbing systems where you have dramatic temporal and spatial variation. And I hope in the next couple of slides, I will demonstrate that a little bit better. All right, so let's just jump straight into a building. So here you have a very simplified image of what you should be expecting with plumbing inside a house. And so a typical house has about 50 meters of pipe length at a diameter of 1 to 2 centimeters. That gives you approximately 30,000 square centimeters of pipe surface area and approximately 15 liters of water if you exclude your boiler volume. And the long and the short of that is it means you have a very high surface to volume ratio. And that means that the biofilms become more and more important in these systems. And that's one of the reasons why we look in detail also at biofilms and not just at water samples in building plumbing systems. And then a point that I really want to make is that building plumbing systems create really unique niches for microbes to develop. So just think about this for a second. So your cold water comes into the building, and then you pump this through your boiler and it heats up to a temperature which you have selected, but that would be a really high temperature typically. So you change the environmental conditions inside that water. And then from there, you pump it to your shower. And in the shower environment, again, there it would cool down. So for example, if the water is stagnating, you're not showering, it cools down and it creates a different environment. Same for the cold water. It comes into the house, it stagnates when you're not using it. So it warms up to your room temperature, and that creates a different environment for the bacteria. You then mix it in the shower together with your hot water, creating yet a different environment. Also, there's different stagnation times in different areas of your house. You have different flow rates, you have different pipe materials. All in all, what I'm trying to say here is a house which we view as a single unit is actually multiple sub-environments. And we know from ecology that if you create different environments, you're going to select for different microorganisms. That is a fact. And so that brings me to the first actual point that I want to make, and that is that building plumbing changes the microbial composition of drinking water substantially. And I want to illustrate this with a couple of full-scale studies. So this study we did approximately 10 years ago, and we looked at 10 real households where we collect cold water from the taps. And we specifically took the first liter, which was stagnant overnight, and then we flushed the water for 5 minutes. Now, what this means is the first liter is approximately 4 to 8 meters of piping, including the tap environment. So you can call this a peripheral sample, but it's quite a large sample relative to your house. And then the flush sample, that is, of course, 20 to 50 meters of piping. So that would usually represent water which is close to network quality water. Now, if you compare these, and this graph shows you the flow cytometric total cell concentrations. So simply how many bacteria were in these samples. You can see that there is a significant difference between the number of bacteria in the stagnant water samples and the number of bacteria in the flush water samples. So if bacteria stays overnight in your house, they are more the next day. And then what we did is we looked at the microbial communities. And for those of you who don't recognize this is a beautiful DGGE plot, which was such a fantastic tool back then. And what you can see from this DGGE plot is that your flushed communities look clearly different than your stagnant communities, again, clustering separately from each other. So a simple take home from this is that overnight stagnation in a building, increase the cell concentration in the water, and change the microbial composition of the water in your house. So then we followed this up a few years ago with a study where we looked at 15 real households again. In this case, we took shower hoses, identical shower hoses, and we installed them in these 15 houses for 1.5 years, let the people operate in the way they normally do. Then we took them out and we collected water samples and we collected biofilm samples and we compared them with each other. And so one important point here is the cold water and the hot water samples that we took. These were really the flushed cold water and hot water. The stagnant water sample was the water which was stagnant inside the shower hose, and then we collected the biofilm from that as well. And so on the plot here on the left-hand side, you just see, let's call this in a simple way, the diversity of the different communities. So the number of OTUs or the number of tox are observed in these different water samples. And what you should see from this is very simply that when your cold water contains the most diversity, once that cold water passes through your boiler, you see much less OTUs. Once you go through the tox into the stagnant water, you see even less. And that is clearly explained by the biofilm in the stagnant environment, which by far has got the least number of OTUs. So the lowest diversity found in the biofilms in these pipes. And I will come back to this in a bit to explain this again. You can also look at this with a classic NMDS plot, and you see the same community shift. So the cold water on the right-hand side of this plot, and as you go through this hot water, then the stagnant water in between, and the biofilms all the way to the left-hand side. Now, a couple of take-home messages from this. So the first is that what you see very nice in that left side image, your warm water changes the composition of the cold water as it came in because it is a different environment. The second point in this is really important is that the biofilms inside our houses do not reflect in any form or manner the drinking water that is coming into our houses, so the cold water as it comes in. And so that is important because we do a lot of our monitoring outside. We don't often monitor inside houses and understand how this change can happen and how it affects the water quality. And then finally, what you should see in this slide, and again, I'll come back to this, is that even though we had 15 identical shower hoses, they developed different microbial communities because they were in different houses exposed to different users and different water coming into them. All right, and so that brings me to the second main point I want to make here, and probably one of the key messages of my presentation, that is that synthetic plumbing material influences biofilm growth and water quality. And that is important because worldwide, there is a clear push towards more synthetic materials being used in building plumbing systems, and often materials with less and less quality control on them. And so what happens if you install a new pipe in a building? So imagine you installed a new shower hose, for example. The first thing is that this new material is going to be exposed to your drinking water, which has got large taxonomic diversity, large metabolic diversity. And these bacteria will then attach to the surface, let's say relatively randomly, and this attachment happens very quick. So that's in the first minutes to hours, you will have random attachment on the surfaces. The second thing that will happen is you're going to have migration from the plastic material into the water phase. And this is going to depend on the quality of the material. So if you, in this example, let's say we have this yellow quality material, it migrates a low amount of organic carbon of a certain type, and the bacteria will utilize this, they will grow, and certain bacteria will dominate as a result, and that will affect your water quality. If you have a different material, let's say material B, some low quality PVC piping, migrates more organic carbon, migrates a different type of organic carbon. Again, this will select for specific microorganisms that can dominate and grow and affect the water quality. And again, I will hope to show some real examples of this later. And so you can say, look at this, and you can say, well, do you really see these different materials in a building plumbing system? And as you can see from this example over here, this is not even a question. You would be absolutely flabbergasted by the large amount of different materials that are used in contact with your drinking water in a relatively small space. So here in this example, approximately 7 different materials just in this small area of plumbing. Now, you can look at this and you can say, well, that is just a worst case example, which you are selecting here for a nice presentation. I will show this as the counter example. So if you take a tap like this, and if you have nothing better to do this weekend, I suggest you do this. You take a Dremel tool to it, you cut the tap open. What you are going to find is at least 8 to 10 different materials in contact with your drinking water in the very last meters, if not centimeters, before you are exposed to it. And this includes problematic materials such as low quality EPDM sealing rings or these flexible PVC hoses, which really migrates a lot of nutrients. Now, looking at this, you say, well, okay, but for example, these are really small materials. Can they affect the water quality that much? I would say yes. So if we focus just, for example, on that grid, which you have over there, and you would zoom in on such a grid, you would find the most spectacular biofilms growing on them. You can look at this a little bit closer. So this image is a 50 by 50 micrometer image. I count roughly, just in a quick count, approximately 100 to 200 cells in this image, which would imply approximately 1 million bacteria per square centimeter of material. So really a nice amount of bacteria. They can influence your water quality for sure. All right, so the point I want to make, so in the earlier slide, I said, your house as a larger system has got these different environments, which is a result of how we build and operate our houses. If we throw on top of this, these different materials, you see that we can even have micro niches and micro environments inside buildings, which will develop even different microbial communities. And so now I would like to show you a couple of actual experimental examples of what I've been talking about. So pilot scale studies that we have been doing in the last few years. In this specific study, we looked at 6 different pipe materials. We exposed them to 1 daily flush. And so this daily flush resembles 1 shower per day. And we did this over a couple of months. Now, the different materials are, for the purpose of my presentation, not really important. They're just all typical shower hose materials. And so what you see on this slide on the right-hand side, this was, if I remember correctly, after 8 months or so, we analyzed the number of bacteria in the biofilms of these different hoses. Take note that this is a log scale. And so what you see is between these different materials, these different pipe materials, there are substantial differences in the number of bacteria that you find in the biofilms. And so very clearly here, your consumer decision to pick a certain hose and the engineering decision by whoever decided which components goes into that hose clearly affected the number of bacteria that will be colonizing your shower hose in your house. Now, you would look at this and you say, well, as long as they stay in the shower, that's not my problem. If they're in the biofilm, that's fine. And so we looked specifically at this and we asked the question, would this also affect the water quality? And so what you see on this image on the left-hand side, this is now the number of bacteria in the water coming out of these hoses as a function of stagnation time. And so stagnation time, how you should view this is, if you are showering only once a day, that means you have 24-hour stagnation time in between. If you're a twice a day showering person, then maybe 12-hour stagnation time in between. And what you see very nicely in this specific example is a very clear correlation between the number of bacteria in the water coming out of these hoses and the amount of biofilm that was on these hoses, and also as a function of stagnation time. So the longer you stagnate, obviously a bit more bacteria ends up in the water phase. So again, just very clearly here, both engineering decisions and consumer decisions influencing the bacteria that you are exposed to as a consumer at the end. And so, as a result of this, you end up having these beautiful biofilms inside showers. Again, I'm not trying to discourage anybody now from taking a shower. In fact, quite the opposite. You should keep on doing this. But the point is very clearly that we need to understand better how these biofilms develop inside showers and inside building plumbing systems in order to be able to control them. Now, that study was really interesting, and that led us to a second pilot scale study where we wanted to understand a little bit better what is happening in detail inside such shower hoses. And in this experiment, as you can see, it was the same shower hose material, and it was aligned horizontally. We showered it 2 times per day for 18 months, and then we analyzed the biofilm in real detail. And we specifically looked, in this study, we specifically looked at heterogeneity on a spatial scale in these biofilms to understand that better. And you might look at this and say, well, that's, it's a meter. How much do you expect? I would argue if you think in terms of the size of a bacteria, which is, let's say for this example, 1 micrometer in size, then a meter represents approximately the distance from Pittsburgh to Denver, if you think on that scale. So you would be expecting that there might be some differences along this space in a shower hose. And we thought that would be worthwhile to investigate this in a little bit more detail. And so what we did is we took the shower hose, we sliced it up into 100 small pieces of 1.2 centimeters, and then we dissected it another time so that we have a top and the bottom, because we had this horizontal alignment of the hoses. And then each and every one of these pieces we analyzed with optical coherence tomography to look at the spatial topography of the biofilm. We analyzed it with flow cytometry to measure the number of bacteria, and we sequence it to see which bacteria are where in this biofilm. And so this first image, I'm just showing this because it's my most favorite image, and it came out really beautiful. So this is an OCT image. It's 1.2 centimeters of a shower hose, and you really beautifully see the biofilm in there. You see a nice heterogeneous structure, a dense biofilm, both over length, and you see a little bit of variation in the structure. Now, what you can do is not only just appreciate the nice image, you can actually quantify biofilm thickness from this data. And so if you stitch all these images together for the entire shower hose, you can get the spatial topography of your entire shower hose visualized, which is what you see over here. So this is the entire 1.2 meters of shower hose. And so what you see then in this image is, you see there are some clear differences. So in the biofilm, so the thinnest and the thickest part of the biofilm has got about 10-fold difference in it. And then really importantly, in this specific image, is that you see that the top of the biofilm is significantly thinner than the bottom of the biofilm. Remember, we have a spatial orientation like this. And so that really got us interested, and we asked the question, why is that? And so I would say there's 2 rough explanation that comes to mind first. And that is that during stagnation, what you would be expecting in the times when the water is not flowing is that particles in the water would settle down and bubbles in the water would rise up to the top. And that was actually possible to observe that as well. And the consequence of that would be is if you have the air bubbles at the top of the hose and you start the flow again, that would create a shear along the top of the hose and shearing off some biofilm, thus making the biofilm thinner. If you have particles settling at the bottom, that would just build up there at the bottom and thus make the biofilm thicker at the bottom. And so that's really interesting. And we thought, okay, we should try and figure out which one of these it is. Now, if you look at the cell concentrations, and again, this was measured every single piece with flow cytometry and then stitched together in this image for the entire shower hose, you see the differences between the minimum and maximum concentrations are not as much as what we've seen in the thickness. And then what is very clear in the image and very important is that the median thickness at the top and the bottom of the shower hose was exactly the same. So we did not observe the differences that we have seen with thickness in the cell numbers. And so if we go back to the cell concentration argument that I've made over here, what I would think from that data is that the air bubble theory at the top is probably not correct, because if the air bubbles were shearing off biofilm, then it would be shearing off cells as well. And so I would expect lower cell concentrations than at the top compared to the bottom. So I suspect most of the thickness differences that we have observed was a result of particles that settled down during this 1.5 years at the bottom and just helping to build up the biofilm from the bottom up. And then the final thing that we looked at in these biofilms was the community composition. And so again, we sequenced every single piece and then looked at how this looks over the entire hose. And the point which I want to make here, really the main point here, is this water that comes into the shower hose contains about 100,000 bacteria per milliliter, somewhere between 2,000 to 5,000 taxa of bacteria coming into the hose. What you see here is that 60% of the entire community of the shower hose was dominated by just 3 taxa. And that is really important because it shows tremendous selection that has occurred in these biofilms. Now you do see differences. So you see differences in the top bottom in terms of the microbial communities. You see spatial differences where one community would be more dominant and less dominant along the length of the hose. But overall, I would say it actually looks relatively similar, but just very, very low in diversity in these hoses. And the explanation for this, I think, comes back to that slide I showed much earlier, and that is that during your initial colonization, I think that is relatively random. And I think the migration of organic carbon out of the hose is probably quite uniform. But then specific bacteria would be able to consume the organic carbon that is migrating. They would find this as a good nutrient source, and they would start to dominate. And then eventually you have a system dominated by certain organisms, which lowers the diversity quite dramatically. All right, and so that brings me to the final part of the talk, and I just want to quickly talk about some lab scale experiments, because all the full scale work and all the pilot scale work is extremely fun and interesting, but it often still is very complex, and it's very difficult to understand some basic principles when you look at that. And so we went down to lab scale and said, can we recreate microcosms where we can study some of these things in a little bit more controlled environment? And so this specific experiment was a very nice, simple and straightforward experiment. So we took these small glass microcosms, we took EPDM coupons. So EPDM is a plastic used for sealing rings. We then took 5 different waters in different experimental jars, obviously, and we added this to the EPDM coupons, and then we incubated it for 2 weeks. And we allowed the biofilms to grow and develop. And then after 2 weeks, what we did is we scraped off the biofilm. We made new coupons, so new microcosms with brand new coupons, and we inoculated this with the old biofilms. And again, we left it for 2 weeks to just grow and develop a new biofilm. And so these are relatively young biofilms grown in 2 cycles of 2 weeks. And at the end, we sampled the water and we sampled the biofilm from the microcosms. Now, what you should see in the image on the left-hand side is very simple. So the orange bars, those were the original water samples. The black bars were after the 2-week incubation. To make a long story very simple here, you see very clearly that irrespective of which water we used, there was a lot of growth occurring. So you have much higher cell numbers occurring after these 2 weeks. And that makes sense because the EPDM bleeds a lot of organic carbon, so there is nutrients for the bacteria to grow. If you look at the microbial community, so in this NMDS plot, so the communities, the original waters are the ones on the right-hand side with the circles around them. And then the biofilm communities are these ones clustering on the left-hand side of the NMDS plot. And again, a take-home message, very simple from this, is after 2 weeks of incubation, sorry, two times 2 weeks of incubation in these microcosms on EPDM, you have a substantial amount of selection. You have a very different microbiome as a consequence. And then another really important point. So these microcosms were absolutely identical, incubated identical, the same pipe material. However, they were exposed to different waters. So they were inoculated with different water samples. And what you can see very clearly on the left-hand side of this plot is that the microbial communities were not identical. So different microcosms had different communities in them. That is the same as what I've shown you in the earlier slide today, from what we've also observed in the real shower hoses from real buildings. And then just if we look again at the diversity, so on the left-hand side, what you see here is the number of taxa in the original waters. So there's the 2 bottled waters on the left-hand side. So they always have very low numbers of taxa anyway. But then the tap water and the groundwater has got nice high numbers of taxa, typical for our drinking water. And this is before we expose them into the microcosms. After we've grown them, if you look at the biofilms, you can see on the right-hand side, you have all of them down to about 30 to 50 taxa. So again, very clear take-home message is after biofilm growth on these synthetic materials, your diversity decreased dramatically in your biofilm. And this, if you paid any attention, but this is on YouTube later, I hope so you can go back if you didn't pay attention. So you would remember that was also exactly what we've observed in real households as well. So that the water that comes in, so your starting water, after this has been exposed to the house and grown in a biofilm, you have much, much lower numbers of taxa, much less diversity in these biofilms. And this is the final experimental slide I want to show on this, and that is just to give you a feeling of this change in diversity that I've been talking about. So this, I believe, was the groundwater sample, came in with approximately 2,000 taxa, relative even distribution, and then a couple of organisms are dominant in this sample. After growth in this experimental setup that I've shown you, you can see a complete shift in the microbial community composition, now down to about 50 taxa in the total sample, and this aquabacterium dominating, which was hardly detectable in the original water sample. So again, take home from this is growth on specific materials, going to reduce the diversity and going to select for very specific organisms. All right, so that brings me to the end of my presentation. I hope that I've convinced you that materials in contact with drinking water and the microbiology that links to that is an interesting, it is a relevant topic. I think I've shown you very clearly that the way we construct buildings and the way we operate them creates multiple microenvironments inside these buildings where different types of organisms can grow and dominate. I've shown experimental examples which show very clearly that our building plumbing changes the biology of the drinking water which is in the network, and so thus, which means we are exposed finally to a different microbiology as what is regularly tested in our distribution systems. In this regard, I cannot argue this strong enough. So different materials have a tremendous impact on the biofilm composition, and as a consequence also on the water quality, on the types of microbes we are exposed to. And very few species dominate on these synthetic materials. So you see a very clear selection on these materials. And then in the last slides, I also argued just again that the processes of dispersal and selection are so important for biofilm formation in building plumbing systems. And hopefully this is information that it needs a lot of development still, but that eventually we can leverage this to construct buildings better and smarter in order so that we have some control over the microbiology of our building plumbing systems. So with that, I've come to the end of the presentation. Acknowledgements to some great people I have worked with and are working with together at the moment. And I end with my most favorite image of a slaughtered rubber duck. Thank you very much.